Hello guys, welcome to this presentation for Biology 2016 paper 2. The specific I'm going to answer the enzyme question, or the question which is coming from enzymes. So stay tuned as I'm answering the questions. Right, so question is saying figure 2.1 shows the effect of pH. So they're saying the effect of pH on the rate of enzyme catalyzed reactions K and M. So we have K and M. These letters, they are representing the reactions. Okay, so one of the things that you need to also to consider when they bring a question under pH, which is the effect of pH on the rate of enzyme catalyzed reaction, the first thing you need to think of the pH scale. So the pH scale is just an instrument which measures the pH of the substance. Now usually a pH scale ranges from 0 up to 14. So it ranges from 0 up to 14. Then the pH of 7, here we are going to have 7. The pH of 7, it is going to be called as neutral pH. It is not going to be acidic or alkalinity or alkaline. Then the pH less than 7, it is called acidic. So on this side here, we are going to have the acidic pH. That is the pH less than 7. Then the pH more than 7 or greater than 7, we are going to have the alkaline or alkalinity. So that is one thing that you need to know here on this part, which is going to help us to answer all these questions. Now, when you talk of the effect of pH, there are some enzymes which work well under acidic pH, and there are some enzymes which work well under neutral, and those that work under alkalinity or alkaline. Now, one thing that you need to know is that an enzyme that can catalyze the reaction under acidic cannot catalyze the reaction under alkalinity or neutrality. That is one thing that you need to know. Right, so let me just give you also an example of the region in the alimentary canal where you are going to find acidic region. So an example of the region in the alimentary canal where you are going to find acidic region is going to be the stomach. Why are we saying the stomach? Because in the stomach, that is where now the hydrochloric acid is being produced, okay? So the walls of the stomach produces hydrochloric acid. So even the enzymes which work in the stomach, they catalyze the reaction under acidic, like protease, pepsin, then those things. Right. Then apart from that one, let me also give you an example of a region in the alimentary canal where you're going to find the alkaline region or alkalinity region. So that region is going to be the mouth or the duodenum. So in the duodenum, all the enzymes that catalyze the reaction in the duodenum, they work under alkalinity, or they operate under alkalinity. So as you can see, this graph here is increasing starting from 0 up to this point here, somewhere there like 2.5, somewhere there. Now beyond 2.5, the graph become narrower. Now we need to know the reason why the graph is becoming narrower here. And remember here we are talking about the pH concentration. That is one thing that you need to understand. So as the pH increases, you are going to find that the rate of enzyme also increases. Now there will be a point whereby the enzyme are going to work at the best, and that point we are going to call it as the optimum pH. The reason why we are calling this one as the optimum pH, this is because we are talking about the pH concentration. If here we are at temperature, meaning we would have said this part J is going to be the optimum temperature. Now this one here we are talking about the pH concentration, meaning J is going to be the optimum pH. Now beyond the optimum pH, which is beyond J, an enzyme is going to undergo what we call denaturalization. So when we talk of denaturalization, it is just the process by which an enzyme or the active of an enzyme is disturbed. So when we talk of an active of an enzyme, suppose if this one here is the enzyme. So the enzyme has two parts. This part is going to be called as the allosteric site, and this part here is going to be called as the active site or inactive site. So on this part here, that is where now the substrate bind. So when an enzyme undergoes denaturalization, meaning this shape here of an active site is going to be disturbed, or the shape of an active site is going to change. So once it is changed, you are going to find that if here we had a substrate which is looking something like this one here, meaning if the shape of this active site is changed, this substrate is not going to be binded on an active site. That is what usually happens when an enzyme undergoes denaturalization. Right, so similarly on this part here, as the pH increases, you are going to find that the enzyme reaction is also going to increase up to the pH of 11. Now, at this point here, we are having L. So L is going to be the optimum pH, because we are talking about the pH concentration. So it is going to be the optimum pH. Now, beyond L, which is the optimum pH, similarly, an enzyme is going to undergo denaturalization. Now, one thing that you need to understand here is that this graph here is more like the pH scale, or it is just representing the pH scale, because it is ranging from 0 up to 14 here. Meaning, the reaction of K here is catalyzing under acidic pH. The reason why I'm saying it is catalyzing under acidic pH, because I was saying the pH less than 7, it is called the acidic pH, and the pH more than 7, it is called alkalinity pH or alkaline pH, meaning M is catalyzing the reaction under alkalinity pH or alkaline pH. So since we have talked of these parts, which is L and J, so these L and J, they are called the optimum pH. Let's try to identify the region in the alimentary canal where reaction K and reaction M can work. So since the graph for reaction K is catalyzing under acidic, meaning this one can work under what? Or it can work where? It can work in the stomach, because in the stomach, that is where you are going to find the acidic region, and the stomach walls, they produce the hydrochloric acid. Then when you talk of reaction M is working under alkaline pH, and I've given you examples of the region in the alimentary canal where you're going to find the alkaline pH or alkaline pH, that is in the duodenum or in the mouth. So reaction M can work well in the mouth or in the duodenum. So let's try to answer some questions. So we have question A, which is saying what term is given to pH at J and L? So at J and L, so it is called the optimum pH. Now you need to be very careful. Some students, they find it very difficult to identify which one is the optimum pH and which one is the optimum temperature. So for you to identify if J and L is the optimum temperature or if it is the optimum pH, the first thing you need to look at what you are having in the x-axis here. So in the x-axis, we are having the pH concentration. That's the reason why we are saying this one is the optimum pH. If in the x-axis we had the temperature, meaning J and L would have been the optimum temperature. So the correct answer here, you can just say the optimum pH. Then B, explain why the rate of reactions K and M goes down after point J and L respectively. So they are just asking us the reason why this reaction of K is decreasing, even the reaction of M is decreasing after passing through L, and this one after passing through M. So we need to explain that. So we can say this is because beyond J, we know that beyond J and L, enzyme or an enzyme is going to undergo denaturalization. So that is what is happening here. So we are just going to say because beyond J and L, an enzyme or enzymes are undergoing or is undergoing denaturalization. So that's the answer. Let's see the next one. So the next one, which is C, is saying name the regions of the alimentary canal where reactions K and M take place. So we have K and M. So as for K, it is operating under acidic. So we can say it is in the stomach. Then as for reaction M, it is operating under alkalinity. So we can say it is just the duodenum or the mouth. So the answer here, we are going to say, as for K, it is going to be the stomach. Then as for M, it can be the mouth or the duodenum. Then as for D, Roman numeral number 1, suggest one enzyme which can catalyze reaction M. So reaction M, it is just operating under alkalinity, and you have given two examples of the region in the alimentary canal where reaction M can take place. That is in the mouth or in the duodenum. So now in the mouth, that is where the digestion of starch or carbohydrates starts. Now, that one happens when the salivary gland secretes the salivary amylase. That salivary amylase catalyzes the what? The carbohydrates or the starch. So you can say salivary amylase or just amylase. Right. Roman numeral number 2. State one food nutrient which can be catalyzed in reaction K. So reaction K is operating under acidic, and an example of a region where you're going to, an example of a region in the alimentary canal where you're going to find the acidic region, it is in the stomach. So an example of an enzyme or of a nutrient which can be catalyzed in reaction K, we can just say the proteins, because the proteins are catalyzed by the protease, and protease work under acidic condition or acidic medium. You can say proteins or protein. Then as for the last one, which is E, state 2 factors other than pH which affect enzyme activity. So we need to exclude the pH. So the first one, we are going to talk of the temperature. Apart from the temperature, we can also include enzyme concentration. Apart from enzyme concentration, substrate concentration, also enzyme inhibitors and enzyme cofactors. So those are the answers, and if you are new to this channel, subscribe and share this video to your friends. Thanks for watching. See you next time.